Hello, my name is Sophie Bicey, my pronouns are she, her, and I'm a PhD student in the Plant Biology Department and Ecology, Evolution, and Behavior program at Michigan State University. Today I'm going to be talking about my project looking at population structure and trait loss along an elevation gradient, working with Drs. Emily Josephs and Jeff Connor. And so to start, I want to introduce trait loss with the example of the cave fish. If we compare these two images, we can see that the cave fish on the top has undergone trait loss when compared to the surface fish on the bottom. Noticeably, there's been a loss of pigmentation throughout the body and eye degeneration. And there's multiple mechanisms of trait loss. The first is direct selection against the trait. In the case of eye degeneration, there's evidence that producing the eye structure takes a cost, but the benefit of the eye structure has been lost in the cave environment when compared to the surface environment. And so there's direct selection against producing the eye structure in cave fish. There can also be correlated responses to selection on other traits. If trait A and trait B are genetically correlated, then direct selection on trait A results in indirect selection on trait B and trait loss. And finally, we can have trait loss due to genetic drift. And this is the typical drift hypothesis that we think about. And this especially can occur when there is founder events, as would occur as surface fish colonize caves, and with small populations. But sometimes there are non-functional traits which are not lost, and so there's evolutionary forces that constrain trait loss. And while there are multiple, today I'm going to be focusing on genetic drift and a lack of genetic variation. And genetic drift is strongest in small population sizes, and can be stronger when there have been founder effects and when there's minimal gene flow between populations. And this way of thinking about drift is opposite of the typical drift hypothesis. In this case, because of the lack of genetic variation, the variation to lose the trait does not exist. And so to imagine our hypothesis here, if this is our elevation gradient, and we start with a large population at low elevation, then there's colonization events up the slope, and the higher elevation populations are smaller than the low elevation populations. And so this low elevation population is larger and it has less genetic drift, which results in more genetic variation and more trait loss. But this high elevation population is smaller, and so there's more genetic drift, resulting in less genetic variation and less trait loss. And the trait I'm going to be focusing on today is short stamen number. The Brassicaceae family has 4 long and 2 short stamens. In this image of Arabidopsis thaliana, we can see 2 long stamens and 1 short stamen. In outcrossing Brassicaceae, like weedy wild radish, the short stamens function to reduce pollen depletion and even increase seed siring success. And the difference between long and short stamen length is maintained by selection. But in selfing species, the function of short stamens is much less clear. As we can see in this image, the long stamens are depositing pollen at the stigma, but the short stamen is depositing pollen along the side of the style. And so my project uses Arabidopsis thaliana because it's a highly selfing annual, and because previous work by Ann Royer and colleagues has shown that the short stamen number in Arabidopsis is a non-functional trait, and that natural populations of Arabidopsis thaliana have trait loss currently occurring. And so here we can see the natural variation in short stamen number that was observed, and it varies from 2 short stamens to 1 short stamen in the middle image and even 0 short stamens, but there's always 4 long stamens. And this variation in short stamen number exists between flowers on the same plant, and the mean short stamen number of a plant differs between plants and between populations. And so my study uses 61 natural genotypes from 16 geographically separated populations along an elevation gradient in the Spanish Pyrenees. In this map, you can see the geographic location of my populations, and they're colored roughly according to increasing elevation. So these red points are the 4 lowest elevation points, and then the yellow points, green points, and finally these blue points are the 4 highest elevation populations. A previous student in the Connor lab, Sam Perez, did the Chamber Common Garden experiment to grow replicates of each genotype and phenotyped them at multiple time points through flowering. And this resulted in a set of about 61 flowers per population. And in addition to the phenotyping data, we have whole genome sequencing, which after SNP calling and some quality filtering, resulted in a set of 1.9 million SNPs. And so the first question we wanted to ask about this data set is, how does mean short stamen number vary along our elevation gradient? And what we find when we look at mean short stamen number by elevation, when each point in this figure represents a population mean, and in the legend you can see that they're colored according to the same scheme in the map, but you can also notice the exact elevation noted. And we see that there's a positive correlation between mean short stamen number and elevation, which tells us that low elevation populations have more short stamen loss than high elevation populations. And this fits with our hypothesis that I outlined in that cartoon example to start this presentation. Our next question was to investigate the population structure in our data set. And we did so through genetic principal component analysis. Here we can see PC1 along the x-axis explains 10% of the variation, and PC2 along the y-axis explains 7.5% of the variation. And we notice that there's an increasing trend with elevation, PC1, and PC2. So in this figure, each point is a genotype, but they're colored according to the same elevation scheme that I've been using. And so that correlation or association between elevation and the principal components tells us that population differentiation is associated with elevation, and it suggests that there might be isolation by distance. We also have an outlier in this green square population, which is the BOS population. And if we jump back to our map, the BOS population is here. And a larger analysis by Castilla and colleagues looked at Arabidopsis thaliana populations from across the Iberian Peninsula. And after structure analysis, they found that this BOS population belongs to the northwestern cluster, while the remainder of the populations in my data set belong either to the northeastern cluster or are mixed. And so we can see that our results agree with this larger analysis. And so to return to our hypothesis, we then needed to look at genetic variation. And we did so through pairwise nucleotide diversity, or PI. And so PI is a measurement that looks at nucleotide differences between genotypes within a single population, and it incorporates the SNPs as well as the non-variant sites that were sequenced. And so here we can see PI by elevation, and each point is that population value colored according to the same elevation scheme. And we have a rather strong negative correlation between nucleotide diversity and elevation, which tells us that the high elevation populations have less nucleotide diversity than low elevation populations. And this pattern is consistent to what we would expect due to drift, as the high elevation populations are smaller, so they have more drift, and the low elevation populations are larger and so potentially have less drift. And to really answer our question, we then need to correlate the nucleotide diversity directly with our phenotype of mean short stamen number. And when we do that, we can see that there's quite a bit of variation between populations that have little short stamen loss in terms of their genetic variation. But we do see that there's a negative correlation, even though it is weak. And so the negative direction is what we would expect based on our hypothesis, because it indicates that when there's little short stamen loss, there's less genetic variation. But when there's more short stamen loss, there's more genetic variation, which is what we had expected. And so in conclusion, the short stamen number is correlated with an elevation gradient, and there's some support that the short stamen loss is constrained by a lack of genetic variation in high elevation populations. And future work on this project will investigate the genetic basis of short stamen number differences. And so with that, I'd like to say thank you to the Josephs Lab and the Connor Lab for their support throughout this project, as well as to Michigan State University for their funding. My email is in the bottom corner here, and you can feel free to reach out to me with additional questions, but I'm looking forward to some discussion in the chat and the Q&A session.